Hello everyone, in this video we are going to discuss the paper Scaling Vision Transformers to Gigapixel Images via Hierarchical Self-Supervised Learning from researchers at Harvard Broad Institute, Bill and Melinda Gates Foundation, and University of Toronto. The authors of this paper use vision transformers to develop models that learn from whole slide images, which are images used in histopathology clinical practice. This type of images can be as large as 150,000 by 150,000 pixels, and unlike most natural images used in common computer vision learning tasks, it has a hierarchical structure to its visual tokens, with patches of size 16 by 16 pixels representing cells of size 1000 by 1000 pixels, capturing important larger-scale characteristics of the images. For this reason, a hierarchical architecture that learns from different image resolutions can be useful for understanding these images and improving performance on downstream learning tasks. For the hierarchical transformer architecture, which they call HIPT, Hierarchical Image Pyramid Transformer, the authors use two levels of self-supervised learning and pre-trained their hierarchical transformers on a public dataset of about 10,700 whole slide images from 33 cancer types. They then evaluate their architecture on 9 cancer survival prediction and subtyping tasks and show that their models based on hierarchical pre-training outperform state-of-the-art methods. They also show how their self-supervised vision transformer can learn inductive biases about the hierarchical structure of visual phenotypes in the tumor microenvironment. So this was a quick summary. Now let's look at the details of the paper. In the domain of computational pathology, our aim is to characterize features in large whole slide images that can help with clinically relevant learning tasks. For example, diagnosis. In the case of cancer, important tasks include predicting survival time or predicting response of patients to a particular cancer treatment. Here, you can see an example of a whole slide image. This example is an image of a tissue slide taken from a surgically resected tumor. The patient undergoes surgical treatment, and the excised tumor tissue is then fresh frozen or formalin fixed, paraffin embedded, and later, the slides are stained with hematoxylin and eosin and viewed at a fixed magnification level, usually 20x or 40x, depending on the institution and the scanner. Whole slide images are different from natural images in several aspects. Among them is, as we discussed, the size. They are typically 50,000 by 50,000 pixels, but can go up to 150,000 by 150,000 pixels. And this is one of the main challenges in learning from whole slide images. Another aspect in which whole slide images differ from natural images is that they are typically at a fixed scale. This scale depends on the magnification set by the scanner. To solve the high resolution problem, weakly supervision approaches are usually employed, whereby small size patches are created from a large image. The features are then extracted from each patch, generating a sequence of embeddings. The last step in this multiple instance learning framework is to perform global pooling of the instances to come up with a representation at the slide level. For example, a random set of patches will be chosen, and the mean feature vector of these patches will be then used as the representation for the whole slide. There are limitations to this multiple instance learning approach that are addressed by a hierarchical architecture in this paper. The patch size is usually fixed, which means the models cannot capture features at different scales. Usually, the patches are 256 by 256 pixels, so they can distinguish changes in nuclei shapes that are typical in tumors, and they can identify if a tumor is present. However, larger scale features such as invasion of the tumor into normal tissue or infiltration patterns of immune cells, specifically a type of immune cells called lymphocytes, and the overall spatial organization of the tissue cannot be captured by the small size patches. This is probably why models based on small patches achieve clinical performance on simpler clinical tasks such as tumor subtype classification or tumor grade classification. The larger scale features are useful for more challenging context-aware tasks such as survival prediction, where these features are known to be predictive of prognosis. For example, the spatial relationship between tumor and immune cells is important for survival prediction, and it can be learned by modeling long-range dependencies using transformers as opposed to conventional multiple instance learning methods. So in this work, we see the development of a three-stage architecture of vision transformers whereby a bottom-up aggregation of visual tokens takes place. At 20x magnification, or half a micrometer per pixel, the visual tokens have the following sizes: 16 by 16 pixels, which represent cells and fine-grained histology features, 256 by 256, which represents clusters of cell-to-cell interactions, and then 4096 times 4096 images that represent macro-scale interaction between cellular clusters and tissue organization. So, for example, tumor-infiltrating lymphocytes or tumor distal lymphocytes, which are immune cells at the borders or the edges of the tumor. At this last level, visual tokens are aggregated to form a representation that is learned at the slide level. This approach is similar to an approach used in NLP called HIPT or hierarchical BERT that learns document representations for long documents. Similar to HIPT, here also whole slide images are modeled as disjoint sets of nested sequences. Whole slide image representations are hierarchically related representations which are learned using a self-supervised learning approach. Specifically, they use the DINO framework or student-teacher knowledge distillation for pre-training at the first two levels, which is the level for the 256 by 256 patches and 4096 by 4096 patches. The models based on this architecture are shown to be superior to other multiple instance learning approaches, particularly in more challenging tasks such as survival prediction, as they are more context aware. The supervised learning approach used up to the 4096 by 4096 patch level performs better than weakly supervised methods when they use k-nearest neighbors in the task of subtype classification. Additionally, the self-supervised approach, which is used up to the level of the 4096 times 4096 patches, performs better than weakly supervised methods when k-nearest neighbors are applied to classification of tumor subtypes. Finally, while self-supervised vision transformers have the property of semantic segmentation of a scene layout in natural images, the hierarchical image pyramid transformers, when applied to whole slide images, can learn visual concepts in histopathology with multi-head self-attention that have a scale corresponding to the scale of the transformers. So you will see fine and coarse-grained visual histopathology concepts when visualizing the different attention maps. So now let's go over the architecture in more detail. To understand the architecture, we need to first formulate the problem working with images and tokens in the context of vision transformers. If we have an image of size uppercase L by L, we generate non-overlapping patches of size lowercase l by l that make up the image. The number of patches is the length of our sequence, that is the input to the transformer. Each patch corresponds to an embedding of dimension d that is learned by a transformer. For standard natural images, the image size is typically 256 by 256, the patch size is usually 16 by 16, and so the image will contain exactly 256 patches, and they will be represented by the corresponding 256 embeddings. For a large whole slide image, the number of patches and thus the token sequence length can be larger than 10,000. This makes standard self-attention intractable. Every token cannot attend to all other tokens, and if we use larger patches to decrease the sequence length, we will simply miss details in the image. This loss of detail will degrade performance. To address this, whole slide images are modeled as nested aggregations of visual tokens that recursively break down to cell-level tokens. The CLS token from the first level is the input to the second level, and the CLS token from the second level is the input to the last, with the input sequence length in the first and second levels being 256. This reduces the total number of parameters in the model and reduces the number of tokens by 256 from one level to the next. At the first level, we have a 256-16 vision transformer or cell-level aggregation, which uses a typical vision transformer similar to what is used in natural images. Each 256 patch is unrolled as a sequence of non-overlapping 16 by 16 pixel tokens, followed by a linear embedding layer with added positional embeddings to produce a set of 384 embedding vectors and a learnable CLS token that aggregates the cell embeddings and serves as a representation for the image. The 4096-256 patch level transformer has the same number of tokens, or 256. The CLS output of the first stage is used to tokenize the patches at this stage. The input has a dimension of 384, while the output has a dimension of 193. For the last stage, or region-level aggregation, the sequence length varies up to 256 depending on the size of the image and the number of non-overlapping 4096 pixel regions. Next, we discuss pre-training. For a model of this size, pre-training is important given the number of parameters. Self-supervision is also important here because few datasets in computational pathology have labels, and datasets are usually small in size. The way pre-training is done in this case is using Dino at the first two stages. Starting by pre-training at the cell level, then freezing the weights of the first stage transformer while learning the weights in the second stage of Dino. This is the same hierarchical pre-training approach that is used in HIPT for NLP. Pre-training is not done at the last stage vision transformer, which is the region-level aggregation. Instead, they fine-tune on each task. However, the pre-trained region-level representations still outperform weakly supervised methods and other embeddings in cancer subtype classification tasks, as we will discuss later. Now let's discuss how pre-training is done using the Dino framework. I'm linking the Dino paper in the description below, but I will briefly describe how they use it in this paper. Starting with the patch level pre-training, we have two networks, student and teacher, and the student is trained to match the output probability distribution of the teacher. This is done using the loss function, which is the cross entropy between the outputs of the two networks. The student network is passed 8 local views, which are 96 by 96 crops, while 2 global views, which are 224 by 224 pixels, are fed to the teacher network. This is to learn local to global correspondence, and is essentially what helps architectures like Dino perform well with self-supervision. For data augmentation, they use color jitter, horizontal flips, and for one of the global views, they also perform characterizing. For the region-level pre-training, the architecture is almost identical to patch-level Dino. The difference is at the input. CLS tokens from the preceding stage are arranged in a 2D feature grid. In the patch level, we had 256 tokens arranged as 16 by 16 2D grid. Each element is based on a 16 by 16 pixel patch. This time, at the region level, we have also 256 tokens. They are also arranged in a 16 by 16 2D feature grid, but here each element has a length of 384. For local global crops, they use dimensions of 6 by 6 and 14 by 14. Next, we are going to cover the experiments. For pre-training, 10,678 H&E stained diagnostic slides are used from 33 cancer types in TCGA. These correspond to around 400,000 regions of size 4096 by 4096 pixel patches. The details of the optimizer used, learning rate, and duration of training is in the paper, and there are only minor differences in pre-training between the two different stages. The fine-tuning is performed for 2 types of tasks: first, classification of cancer subtypes, and then survival prediction. For survival prediction, they use a cross-entropy loss, not the more conventional Cox loss. We will cover this loss function in more detail in another video. The HIPT method is compared to other multiple instance learning methods. Here it's important to note that the input features for all methods are the output of the vision transformer at the first level, or the 16 by 16 patch tokens. So what's really being evaluated here is the hierarchical architecture and the recursive pre-training approach. For classification, there are 3 tasks: subtyping breast cancer, subtyping non-small cell lung cancer, and subtyping renal cancer. Additionally, for classification, they do another exercise of looking at the representations learned without fine-tuning. So looking at the output embeddings for each of the two stages, the patch and region level embeddings, and taking the mean of the embeddings to represent each slide. This is also compared to ResNet 50 embeddings, which are pre-trained on ImageNet. These three approaches then are compared in their classification performance using k-nearest neighbors. Both in the case of using k-nearest neighbor with the extracted embeddings, or in the case of weakly supervised learning, HIPT outperforms other methods. For survival prediction, they evaluate their architecture on 6 cancer types, also from TCGA using a TCGA datasets, and HIPT mostly outperforms the other methods, giving superior performance in 4 out of the 6 datasets. Now we want to compare the hierarchical transformer architecture applied to whole slide images with the standard vision transformer applied to natural images, particularly in its semantic segmentation property. So here they analyze the different heads and multi-head self-attention for both the patch level and the region level. For heads 1 and 2 in the patch level, we see stroma and red blood cells attended to. And we see in heads 3 and 4, they attend to white spaces. White spaces can be a lumen, which is an empty structure inside the body, or it can be fat. Fat regions also appear white on histopathology slides, or it can also be air pockets. All of these can be delineated in this figure. For the region level, visualizing attention heads of the pre-trained vision transformer shows more coarse-grained characteristics. In heads 1, 2, and 3, we see the interface between stroma and tumor, and heads 4, 5, and 6 show high tumor cellularity regions, or regions with high-density tumor cells. These heads show patterns of nested tumor growth and invasion of tumor into fat and stroma areas. It's these spatial patterns we see here that are important for complex learning tasks. We can see these attention maps and the different phenotypes they are able to isolate in this figure. The first panel is the regular H&E stained slide. A is a magnification of the top left green box, and it shows a region where the tumor, the dark purple parts, invades muscle and stroma. Mostly the pink parts are muscle. The second, bottom left, is a visualization of the attention head at the patch level. High red values for cells and blue low values for the white spaces. The top right are visualizations of the attention heads for the region level. On the left, we see panel A, the stroma and muscles are red, and panel B is mostly blue. Subpanel B is mostly red on the rightmost panel, while subpanel A here is blue. So this is a visualization of the tumor region. When they factorize the attention heads of both levels, we get an interesting visualization, with hierarchical attention delineating cells within associated stromal tissue on the left subpanel, and a tumor region dense with tumor cells on the right. In conclusion, this work is very important for computational pathology because of the use of self-supervision in the novel architecture that tackles the size challenge of whole slide images. We saw that the model that learns representations that are self-supervised still outperforms state-of-the-art supervised or weakly supervised methods. The authors list some limitations of the study, and mainly they have to do with the use of TCGA data and the lack of other independent test sets that are needed to evaluate the model. Another limitation of using a TCGA data, although it's a large data set, is that the subjects largely have European ancestry. This has implications when using such data sets for pre-training, as it can lead to model bias. It's also important to note that even when using this large data set of thousands of whole slide images, pre-training could only be done at the first 2 stages. They were only able to use fine-tuning for the final transformer stage that learns the slide-level representations. This points to the challenge of data scarcity in computational pathology. This concludes our discussion today. Please subscribe to get notifications when we post new videos. Thank you for watching.